Yes. Hello. So I'm starting this session today. So today we'll be learning about the enzyme inhibition. So the enzymes which do the catalytic activity and converts the substrate into product, it can be inhibited by several chemicals, so that regulates or hampers the activity of the enzyme. So these inhibitors we can divide into two categories. One will be the irreversible inhibitor. Another one will be reversible inhibitor. Okay. So irreversible inhibitor is such kind of inhibitor when binds to the enzyme, it kinds of destroys the functional group of the enzyme. Okay. It destroys the functional group of the enzyme, and thus enzyme will lose its catalytic activity. However, and there is no going back in this case. If the irreversible complex between enzyme and inhibitor is formed, the enzyme cannot be reused or can get rid of the inhibitor. However, in case of reversible inhibitor, this kinds of inhibitor can attach to the enzyme and also is able to detach in several condition and cases, and thus the enzyme becomes reusable for its activity. So as I'm talking about the irreversible inhibitor first, so there are different kinds of irreversible or different irreversible inhibitors which we'll be talking. So first of them is the first I can tell is the DFP, which is diisopropyl diisopropyl fluorophosphate. So this is one inhibitor which is DFP, and it can inhibit acetylcholine esterase enzyme, and also can inhibit the serine proteases, for example trypsin and chymotrypsin. And if I talk about the mechanism of action, so this is bitter. Okay. So the enzyme has such functional groups, for example serine. So serine has a functional group of OH. Okay. CH2OH. So yeah, so here it is. Fluorine. So this is DFP, diisopropyl fluorophosphate. Okay, so this fluorophosphate group can react with the uh, catalytic or the active amino acid, or the amino acid which is present in the active site or responsible for the catalytic activity, and thus it binds to the enzyme in an irreversible manner. Enzyme, and thus now it's a covalent complex has been formed between the inhibitor and the enzyme, and this is a irreversible bond, and that is why it cannot be broken and the enzyme cannot be used for its catalytic activity. Okay. Similarly, this acetylcholine esterase, which is helping in the neurotransmission, it can also be affected by insecticide which is malathion. This can also inhibit this acetylcholine esterase. Okay. So this acetylcholine esterase it converts acetylcholine into the acetate and choline, and that is how it helps in the neurotransmission. Next, we have this inhibitor, which is iodoacetamide. It can inhibit the SH group of enzymes. It can inhibit imidazole group, imidazole group of histidine, histidine, and it can also group inhibit the OH group of serine. Serine. Okay. So other than that, third one is everybody knows cyanide. So cyanide also inhibits the cytochrome oxidase enzyme of electron transport chain etc, and thus uh, affect the ATP production, and thus all the physiological process, downstream physiological process will be affected. So cyanide is also an irreversible inhibitor. Fourth is fluoride. Fluoride inhibits enolase enzyme, and in that it removes the magnesium and manganese ion which is present in the active site or the catalytic site of this enzyme enolase. Okay, so this is what is the other than that, we also have in irreversible category only suicide inhibitors or suicide inhibition, or we can also call them as mechanism based inhibitor. So mechanism based inhibitor because it utilizes the mechanism machinery of the enzyme, and like this inhibitor, it is an with the help of enzyme it catalyzes into a more effective inhibitor, which is a analog of the further substrate, and when this particular one reacts with the enzyme, it forms an enzyme inhibitor complex, and this enzyme literally commits suicide and becomes such non-functional. This inhibitor is more potent inhibitor than the original one. So it utilizes enzymes machinery only to become a more potent inhibitor that can block the activity of the enzyme. One such example of these is allopurinol. So allopurinol, with the help of the enzyme xanthine oxidase, gets converted into oxypurinol. Okay, and now this oxypurinol can bind to the molybdenum sulfate site of the enzyme, which interacts with the substrate, the original substrate of the enzyme, and thus this binds and complex and doesn't produce any product. Okay, so this oxypurinol is a more potent inhibitor formed with the help of enzyme itself, xanthine oxidase, from the allopurinol inhibitor. So allopurinol is also known as anti-gout drug. Okay, so this enzyme xanthine oxidase. So what it does is pages. So from our diet, we purines, we get hypoxanthine, hypoxanthine converted to xanthine, and now this xanthine gets converted to uric acid. Okay, the help of xanthine oxidase, and this uric acid further gets converted urate crystals into urate crystals, which gets deposited in our joints and causes gout disease. So this drug, which is allopurinol inhibitor, which used as anti-gout drug, it can inhibit the xanthine oxidase by a suicidal inhibition activity by help utilizing xanthine oxidase to become a more potent inhibitor oxypurinol, which can affect, which is a state analog of the enzyme substrate, and now it is a more potent inhibitor than allopurinol and inhibits the xanthine oxidase catalytic activity. Okay, similarly we have another anti-cancer drug, which is 5-fluorouracil, gets which is get converted into 5-fluorodeoxyuricylate. Okay, and this can inhibit the enzyme thymidylate synthase, and thymidylate synthase converts in uh, DUMP to DTMP, thymidine monophosphate, which ultimately gets converted deoxythymidine triphosphate and take part in DNA synthesis. Okay, so here, so this thymidine synthase is a folate dependent enzyme which utilizes the methylene tetrahydrofolate and uh, forms the methylation and converts and forms the deoxy thymidine monophosphate and releases the dihydrofolate, which gets converted to tetrahydrofolate. And this cycle goes on. This is the folate cycle. So this is a folate dependent enzyme. So dihydrofolate to tetrahydrofolate conversion is also inhibited by aminopterin, and it can block the DNA synthesis. Mm, and the enzyme which is involved here is dihydrofolate reductase, DHFR enzyme, and aminopterin can inhibit the DHFR enzyme and ultimately inhibit the DNA synthesis. Okay. Next, if we'll see the questions related to it. So the functional group of an enzyme necessary for catalytic activities is destroyed. So the functional group of enzyme is destroyed in case of irreversible inhibition, and all these three are part of reversible inhibition. Next question, if we'll see, is the imidazole group of histidine is inhibited irreversible by. So this is the correct answer, idoacetamide. It can inhibit the imidazole group of the histidine. Then we have the fluoride, which inhibits the enzyme enolase by removing from active site. Which ions? So as we've told, magnesium ion and manganese ion. So manganese ion will be the correct option here. Then we have the following insecticide is the inhibitor of acetylcholine esterase that I already mentioned, malathion. And we have the question that the enzyme that commits suicide by initially activating allopurinol into a transition state analog is. So the enzyme that commits suicide is xanthine oxidase. And from allopurinol, it forms oxypurinol. So the inhibition of enzyme dihydrofolate reductase, DHFR, blocks the formation of. So from dihydrofolate to tetrahydrofolate. It's done by DHFR and it can be inhibited by aminopterin. aminopterin. So the inhibition of enzyme DHFR blocks the formation of, so it will be tetrahydrofolate by aminopterin. So the correct option will be B. So this is we talked about the irreversible inhibition. Now we have the reversible inhibition. So for reversible inhibition, we have three types. First will be the competitive. Second will be, let's say, non-competitive. Third will be uncompetitive. Uncompetitive inhibition. So these are the three types of inhibition. So if we'll talk about this, then here in the competitive inhibition, what happens is it is a in competitive inhibition, the inhibitor binds to the active site of the enzyme. Okay, so we call the enzymes. So suppose this is your substrate, and in case of competitive inhibitor, this is a inhibitor. Okay, so the inhibitor and the substrate are the structural analog of each other. So in case of substrate, so the inhibitor competes with the substrate, and the inhibitor can bind to the active site of the enzyme and doesn't allow the substrate to bind, and that is how it is a competitive inhibition. So if I say, so this is the active site or catalytic site of the enzyme, and this is the regulatory site or the allosteric site of the enzyme. Okay, so in case of competitive inhibition, the inhibitor binds to the active site of the enzyme and competes with the substrate. So here in this case, increasing the substrate concentration will help and compete with the inhibitor and remove the inhibitor, so that the enzyme is now occupied with the substrate. So doing so, the km of the enzyme, km of the this enzyme for the substrate increases. However, there will be no change in the maximum velocity of the reaction. In non-competitive, what happens is the inhibitor, it binds to both the free enzyme and the enzyme substrate complex. Okay, so it can bind to the however, the inhibitor does not bind to the active site of enzyme. So here in this case, the inhibitor binds to the regulatory site of the enzyme, and thus what happens, there will be a change in the substrate, change in the substrate conformation. So that is why the substrate is no longer a fit here. So no catalytic activity is happening. Okay. However, in this inhibitor can also bind to the enzyme substrate complex as well. So already substrate bound. So there also the inhibitor can bind and change the con causes the conformational change here. Okay, and this becomes a non-functional enzyme. Okay, this doesn't show the activity. In this case, in the uncompetitive inhibition, what happens? The inhibitor binds when to the regulatory site only, but when the substrate is bound to the enzyme, then only it binds and doesn't allow the enzyme to. So here in this case, I can say the competitive inhibition, the inhibitor bind to the active site, and non-competitive inhibition, it binds to the allosteric site, and in non-competitive inhibition, it binds to the enzyme substrate complex and inhibit the activity of enzymes. So suppose there is a substrate attached and it needs to release certain, by breaking the substrate release to need to release certain component or one particular component of the substrate. So inhibitor doesn't allow to do that when it binds to the enzyme substrate complex. So that is why this becomes non-functional. Okay, this also not functional. So these are the three types of reversible inhibition happens. So in this case, so in this case, so here there will be no change in the Km because it allows the substrate to bind. There will be no change. However, the Vmax of the reaction gets decreased. In this case, Km and Vmax both shows a decrease. Yeah. So these are the all basic concept about the reversible inhibition. Okay. So for one example of reversible inhibition is uh, the inhibition of succinate dehydrogenase. So this is an enzyme, a big enzyme. So what it does, it binds to succinate and convert it to fumarate. Okay. Okay. So convert it to fumarate. Succinate, this is succinate dehydrogenase. However, this succinate structural analog, which is malonate, it can also, it is a same structural analog, so that is why this can also bind to this enzyme, but there will be no formation of the product. So this is a competitive inhibition example. Succinate dehydrogenase get inhibited by malonate, and which competes with succinate because of the structural similarity, and there will be no product formation in this case. In uncompetitive, what happens? In uncompetitive or non-competitive, so one example of this is an example of this is non-competitive. This one was competitive or non-competitive. Example is so threonine. So it forms isoleucine. It is the example of feedback inhibition. So this isoleucine now binds uh, or sorry, this isoleucine now inhibits this enzyme. It is a feedback inhibition. So this inhibits this enzyme which is threonine dehydrogenase. Dehydrogenase, and it inhibits uh, binds to the allosteric site. So isoleucine binds to the allosteric site of and threonine dehydrogenase and doesn't allow threonine to bind to it, and because it is causing the conformational changes. So, so suppose this is the this is threonine. This is threonine dehydrogenase, which is the enzyme which converts it to further isoleucine. Now we have, so what happens when we have this, the inhibitor, which is isoleucine, when formed enough, it binds to the allosteric site and causes conformational changes. So that now this particular threonine is no able to bind to the threonine dehydrogenase enzyme. So that is why no further formation of isoleucine happens. So this is a non-competitive inhibition example. Next we have another example from the competitive inhibition, sorry, uncompetitive. So in uncompetitive, for example, in non-competitive inhibition, phenylalanine can inhibit the alkaline phosphatase enzyme when it is bound with its substrate. The substrate is bound, and the inhibitor is also binding at this particular time only. It is not allowing the release. So alkaline phosphatase, which causes the release of the inorganic phosphate from the substrate. So the inhibitor, after the inhibitor binding, the release of the inorganic phosphate from the substrate, from the enzyme substrate complex is inhibited. So that is an example of uncompetitive inhibition. Okay. And here is the Lineweaver-Burke plots for the enzyme inhibition. So normally we have the blue one, if we'll see. So here we have the uh, y-intercept, and we have the x-intercept. X-intercept is our Km, and y-intercept is our Vmax in a reciprocal form. And this is for a linear equation, this. So 1 by V naught, slope of this m is will be the slope, will be km by v max, and x will be the substrate concentration, which will be reciprocal of that, and c will be the pi intercept, which is 1 by v max. So this is the new Lineweaver block equation. Okay. And here, when another, so in case of competitive inhibition, you can see the y-intercept remains the same. However, there is a, I think I mentioned, yeah, yeah. So here you can see it is coming closer, that means it shows much more value than the previous value when in presence of substrate. So now the in this case of this, the Vmax remains the same. We can see the slope. However, there will be an increase in the Km value by alpha. So the now new Km will be alpha Kms. And here in uncompetitive both gets decreased by the factor of alpha, and in non-competitive only Vmax decreases and there will be no change in the Km value. So if we see the question, so inhibition of succinate dehydrogenase by malonate is an example of, we just now talked that succinate dehydrogenase is inhibited by malonate, which is a structural analog of its original substrate succinate. So it is a competitive inhibition. Next, we have the type of inhibition observed with inhibitor when Vmax is decreased and Km remain unchanged. So Vmax is being decreased, however, Km remains unchanged. Km remains unchanged when the enzyme does not bind to the uh, active site of the enzyme, and that happens in case of non-competitive inhibition, where we saw here in this case where the inhibitor is binding to the allosteric site. So this is a non-competitive inhibition. Correct option will be this. Then with the type of inhibition where the inhibitor can bind to the free enzyme as well as the enzyme substrate complex. So that also will be the non-competitive inhibition. Next we have the uncompetitive inhibitor rapidly binds to the ES complex and convert into ESI complex and converts it into the ESI complex. Yes. So the Km in the presence of uncompetitive inhibitor, and so Km we know it decreases because, and then inhibitor and it dash the Vmax as well, and it decreases. So both will be reduces. Okay. So decrease in the Km and reduces in the Vmax value as well in case of uncompetitive inhibition. So the inhibitor that change the shape of the active site so that the substrate cannot bind is also a non-competitive inhibitor. Next is dash binds to regulatory site on threonine dehydratase. Yeah, that was a dehydratase, not dehydrogenase. Sorry, I mentioned incorrect previously. So it was a dehydratase and function as this. So that's the example which I have given, which is isoleucine, which binds to the allosteric site or regulatory site on threonine dehydratase and function as a non-competitive inhibitor. Next we have is, this is recognized as a specific inhibitor for a alkaline phosphatase by functioning as a, so alkaline phosphatase I mentioned, phenylalanine, which is an uncompetitive inhibitor because it binds to the enzyme substrate complex. So phenylalanine is the inhibitor, specific inhibitor for alkaline phosphatase and functioning as a uncompetitive inhibitor. So this will be the answer. So these are the questions which I've taken today. Hope it was helpful. So I'll end the session. Thank you.